I probably don't even need to ask for a show of hands. I mean, there's nobody who's completely content with how we're managing seed transfers and regulating transfers throughout the region. This is a source of enormous frustration for everybody that's growing shellfish or producing seed. And what we're realizing is that a framework for managing transfers and managing disease and transfers that was developed prior really to the existence of any substantial aquaculture industry, when there were a few growers here and there that weren't moving anything really around, just simply doesn't work when we have an industry that's worth hundreds of millions of dollars, that's regionally integrated and moving product all over the place across state lines. So we're rethinking the way we think about seed transfers and think about managing these transfers. So, and this is a major area of interaction between shellfish production and regulation, and it's all for the purpose of really, for the most part, controlling these diseases. And dermo disease, which is Perkinsus marinus caused, which is in the illustration on the slide right now. This is a major regional pathogen and a pathogen on the East Coast as well. So this is certainly a key area of interest for regulation, not moving any more dermo around than we need to. But all of the aquaculture that we do is conducted against a broader backdrop of a number of pathogens of these species that we're producing. In the upper left is MSX, or Haplospermium nelsoni, which is present on the East Coast from Atlantic Canada to Florida. QPX in the upper right is a clam pathogen, widely distributed on the Atlantic coast. ROD, or what we used to know as JOD, a bacterial pathogen in the bottom left, occurring in the Northeast. And Benamia is a parasite that's on the radar now. We discovered this in about a decade ago in Chrysostia eriacensis trials in North Carolina. We know it infects C. virginica under certain circumstances here and there. So this is sort of the backdrop of some of the pathogens that we're concerned about. And we really don't want to make these worse as a product of aquaculture activities. So the regulation is basically focused on controlling these pathogens in the context of interstate transfers. And typically the state of play as it exists right now is for regulators at the state level to require health certifications of all batches of product of any size that are proposed for interstate transfer. So this is anything from larvae or germplasm through seed of different sizes to even brood stock that will be moved from place to place. And typically the state regulations with regard to transfers specify that the policies are zero tolerance with regard to the presence of a pathogen. So the detection of any amount of any of these pathogens on the list is enough to halt a transfer across state lines. So the downside of all of this, of course, there's costs to industry and time and money. The analyses that we do, like my colleague in my laboratory, I mean, some of these analyses can take a week or 2 weeks if we're talking about numbers of broodstock samples that need to be evaluated. There's a cost of this to industry, which could be in the hundreds of dollars. Broodstock certifications can be over 1,000. So there's time, there's money, there's a reduced ability to take advantage of sort of rapid, immediate opportunities to move product around and make sales. The benefits of imposing this level of regulation of this type at least ensures that, well, if we're not moving anything around because of these pathogens, we're ensuring that we're not going to make anything worse. We're maintaining a high level of biosecurity with regard to these specific pathogens. Well, this is probably not true either, and this is something that Dave Bushek and I have written about. We sort of know that when regulation's too excessive, when the restrictions to transfer are too onerous, that industry is going to need to move product no matter what anyway. And the product is just going to be moving through different channels that can be less biosecure, that can increase the risk of disease more than the ones that we would sort of like to sort of channel the commerce through. So this can paradoxically have the effect of reducing biosecurity if the regulations are sort of not tuned right to what industry needs and what's realistic in terms of the biology of these systems. So this is something that we recognize as a real problem. We think and the regulators may think they're doing a good job, but they're probably actually making things worse, in a number of ways, complicating industry and actually maybe increasing disease risks to everybody. And again, zero tolerance policies I mentioned before. These are on the books everywhere, and these are clearly unrealistic in our region where we have pathogens like Perkinsus that are not sort of present in scattered locations and can be avoided, but they're present everywhere, infecting, literally every oyster where salinities are above 10 parts per thousand. So we need to have a different sort of model for how we deal with this, and that's what I'll be basically introducing over the next few slides. This is some of our current thinking, and I should say this is an effort. This has been funded by a number of grants. So these are projects that have deliverables and reporting and start dates and end dates, but this is really a journey because we're really trying to change the culture around how we think about managing these diseases, changing regulation at the state level. All of this sort of happens slowly, but things are sort of starting to move. The core group that's working on this is between Rutgers and VIMS, with USDA colleagues. The East Coast Shellfish Growers Association has been very supportive of these efforts, and we've had most of our funding through NOAA and Sea Grant sources. So what are we doing? First, dealing with zero tolerance, which is really such a problem. We're really trying to sort of think about things a different way in terms of how the regulators are dealing with these parasites. And really where we want to go is moving away from zero tolerance, where any level of detection of any of these pathogens on the list shuts down a transfer, to really talking more about how much is too much. We know that there's a lot of natural transmission of things like dermo and on the East Coast, MSX, Haplospermum nelsoni. Moving a little bit of one of these in some small seed is probably not going to materially impact transmission dynamics in a recipient location. So bearing this in mind, can we learn to live with a certain amount of the presence of the pathogen that's present in these shipments, so that we can allow reasonable commerce that's not going to make anything worse to proceed. And we're making some progress in starting to shake regulation off of this zero tolerance paradigm in places like Virginia, Maryland, and North Carolina. Very notably. Part of the solution is not focusing on the presence of the pathogen, but on the presence of patent disease. So maybe we see a couple of Perkinsus cells, but when we look at the histology and the microscopy, these are healthy animals. And we can specify that in our analyses, that, we're detecting a little bit of these ubiquitous pathogens that are everywhere, but these are very, very healthy animals that are going to probably do well when they're transplanted someplace else. So this is a major focus of our effort, is changing sort of the psychology and how people perceive risk with regard to the presence of any of these pathogens. Eliminating regulatory uncertainty. So, Dave and I, Bushek and I published a recent paper where we highlighted a number of paradoxes with regard to managing disease, and we highlighted this paradox of uncertainty, that when regulators are uncertain about the risk of a transfer, they tend to stop things and just say no. And like I'd said earlier, just for regulators to say no to everything because they're uncertain about it, has the potential to actually increase disease risk, because industry is going to find other channels around the process. So it's the paradox of uncertainty. It's sort of, we could call it the paradox of regulatory paralysis, that the regulators may think that this is actually better than an alternative, but it could be worse. So what are we doing on this front? This is an effort that Rutgers is leading to develop a disease database that basically captures, in a web-based form that's going to be available, accessible to everybody, the current and historical status of the presence of shellfish pathogens and the prevalence and the intensity levels up and down the East Coast presently, which is the focus of our efforts, integrating data coming from state monitoring programs, but on some level as well on data coming out of farms, with appropriate coding so that the farms aren't identified to location. For example, if there's only one farm in a water body, we're not going to present that data, but if there are a number of them such that it can be coded, so we're not attributing data to a specific place, that will be in the database if industry is comfortable with that. But highlighting all of this, making this easily accessible for one, industry, so they can know sort of, all right, well, this is my disease status on my farm. South Carolina has a similar disease status, so this might be a place I can get seed or move seed to, because it's a like-to-like transfer with regard to disease risks. But regulation at a glance can see what the current science, the current monitoring says with regard to what diseases are where and at what levels, so they can much more quickly move to that yes that we want them to make in terms of a regulatory decision. So this is something that we're presently populating, and it'll probably be open to the public, not fully populated with data, but probably within a month or so after the Aquaculture 2019 meeting. Another major area of focus is promoting the most biosecure commerce. We know that not all of our commerce, all of our transfers are equally sort of secure, biosecure in terms of pathogen levels. Let's look a little more closely at this. So just this graphic that I presented here, you could look at the wild background line at the top, basically which highlights, that's the levels of disease, in wild oysters, in a natural population. Up the y-axis, from the bottom to the top, the y-axis, that's infection level or disease transmission risk, however you want to think about it. So we might view that as being sort of maximum in the wild animals. Broodstock, which are, effectively, they're out there in the wild, they're filtering pathogens, they have similar disease profiles to the wild oysters, wild clams, if that's what we're talking about. If we look at submarket or market size animals, they can have a range of different infection levels, but they can sort of approximate the same levels that we would see in these older wild animals or broodstock. If we look at germplasm, sperm from tetraploid broodstock that have been out in the water for a while, they can have a range of different infection levels, but also that can be quite high, like the older wild animals in these populations. But if we look at smaller animals, nursery animals that aren't very large, that haven't been out there filtering raw water for very long, hatchery seed that is on filtered or treated water of some level, that's very small, hasn't been filtering very long, these tend to be very, very disease-free products, very biosecure products. So let's see what we can do to try to incentivize transfers in this safest material to be moving. And one of the major efforts that we're pursuing is a hatchery certification process, where we can basically, regardless of the outer, the disease levels, out in open bay, on which a hatchery resides, that we can basically define a hatchery as a compartment apart from that open system that has its own unique disease levels, which tend to be essentially non-existent in typical cases. Let's define that through some surveillance in the hatchery, some data collection, to demonstrate that, this is really very, very minimal or absent in terms of pathogen presence. And then let's reduce the barriers to those hatcheries that are sort of in that program, that have been sharing information, letting us collect data, where we could demonstrate an absence of pathogens, so they can move their product much more freely without having to go through the certification process and the time and money that would take. So that's a major focus, is trying to get hatcheries, get regulators on board with this, and get hatcheries into a program where we could basically streamline their ability to move this most safe product that everybody should be comfortable with. Rethinking pathogen surveillance. So, of course, a lot of what I've been talking about, we do these batch certifications. Any animals that want to be, that somebody wants to move across state lines need a health certificate. So this is regulation at the batch level. What we're doing more of now in the Mid-Atlantic is using monitoring of wild populations. So basically taking the burden off industry, putting it on the monitoring programs that are run by the states and using the monitoring data rather than the batch testing data to inform the regulatory decisions about whether this is a good transfer from A to B. And this is something, for example, that Maryland is doing a lot of. They're using Virginia monitoring data to basically determine what levels in Maryland would have would represent like-to-like transfers. What area in Maryland would have a similar disease profile to, say, the James River in Virginia, and then let the transfers flow more freely from those two areas because they're equivalent in levels of disease. So moving toward trying to really remove the burden from industry, put it sort of back on the states in terms of baseline data collection to try to just streamline things a little bit further. We assume, and we have a lot of data pointing to this, that aquaculture animals tend to have qualitatively very similar disease profiles to wild oysters, if they've been out on a farm for a while, but they tend to be a lot lower because they haven't been out there as long. They're planted sort of as a group, at some physical distance away from sources of infection out there. They tend to have lower infection levels. And all of this is taking us toward sort of defining zones, sort of like this zone has this sort of disease profile. Let's define zones so we could define what like-to-like transfers might be from zone to zone, again, to try to remove regulatory interference in this. So is any of this stuff, so we're pretty optimistic that we're going to be able to make progress on this, even if it sort of progresses sort of slowly. Is any of this a model for biosecurity management in the Gulf with regard to these major diseases? And really, all of it probably is. I think the only limitation in the Gulf area is that we don't have as much baseline data on diseases that we do on the Atlantic coast. We have a ton of data with regard to dermo, and this is the Oyster Sentinel website that I've portrayed up here. A lot of excellent perspective on Perkinsus, but we don't really have good data on anything else in terms of what other pathogens might be present, what the regulators need to be thinking about. So this is really something that I think the Gulf Coast, to try to move forward in dealing with some of the regulations related to transfers and streamlining things, maybe needs to give some thought to try to collecting some more information on. And just as a final note, we think a lot about managing and regulating and controlling these specific pathogens like Perkinsus and MSX, but we really want to always bear in mind that we're trying to take a more holistic view of oyster health and of shellfish health generally. It's not just these specific pathogens that we're concerned about, but trying to understand the contribution of environmental stressors under a changing environment to these diseases, to disease in general. The role of bacterial and viral pathogens we probably haven't fully appreciated because this is really not an area in which we have as much surveillance or expertise, really. Certainly in areas like the West Coast and Europe, we know that there's a lot more going on than we appreciate, than meets the eye on that front. And then finally, understanding these phenomena. We talk about triploid mortality and summer mortality, and there are some real problems that are contributing in a very real way to limiting production and limiting profitability at the farm level. And we still don't really quite understand what's going on with a lot of these issues that may relate, for example, to oyster genetics or ploidy or physiology. But we need to focus much more on trying to learn more about what's going on those fronts too. And with that, that's all I had, and I'm happy to take any questions if there's time. Yeah.
Got any questions for Ryan? Any questions for Ryan?
Yep. Steve? Yeah. So that's that comes up all the time, and Mike Congrove had asked me about that earlier. Like, what about this? We need to be concerned that this dermo might not be the same as that dermo, and so there's an 80% prevalence here and an 80% prevalence there, but it's not the same. And we don't have a really good handle at all on strain level variation in these pathogens, but we don't have evidence. We know that there's going to be some level of variation here and there, but we don't have any reason to believe that there are like material differences in virulence based on any scientific foundations at this point that suggests that we should be managing them based on the unique dermo in South Carolina versus North Carolina versus Louisiana. And our position has has always been let's not. I mean, let's manage based on the science we have, rather than on these theoretical questions that there may be nothing there of consequence with regard to transmission dynamics. I mean, these are good questions, and this really should be the focus of some research, in the Gulf, in the Atlantic. Are there, is there local adaptation to the parasite you see all the time? And then if you disrupt that, or you introduce a novel genotype, is that going to have a totally different outcome? And I'm just so skeptical, given the movement of animals and the pathogens, and the sort of homogenizing of all of that at the regional level. I'm just so skeptical that there'd be a lot of structure there, but that's really should be the focus of some research. Well, so in the Gulf, we're only aware of Perkinsus marinus as a significant pathogen in the Gulf, and there's no evidence that there's anything else. But again, this is I made the point earlier. We don't have as much data as we need, really. Like there could be something someplace, that we, in Texas, that we, because we haven't done any histology in Texas, we may find that, well, there's something there that we don't want to move here. But I mean, we assume that we've looked enough that we're not going to find anything. The question of moving stuff between the Gulf and the Atlantic, in either direction, I mean, this is something that, we've had, nobody's been completely comfortable about that. And if there's going to be movements, that's where, like a very, very high level of stringency with regard to testing. And I mean, nobody's going to move your brood stock from the Atlantic coast to the Gulf Coast. The question would be, look, we can demonstrate that hatchery facilities are completely free of MSX, for example. So then, is that a possibility to move? Because we could demonstrate, very rigorously using the most sensitive tools we have that, look, this is just not present in this facility. After, with years of surveillance, that's still going to be a regulatory decision at the state level, but that's a conversation maybe to have, depending on how far afield someone needs to go to get a seed product.
I would just add to that, LaDon, one of the things that we struggled with here was zero tolerance means that you could have seed that show up that they've got a little bit of dermo that are going from a hatchery in Mississippi and they're trying to go to Florida. And Florida will reject that even though they have dermo in Florida because they have a zero tolerance process. And so that has led to part of the seed crisis that we have is partially because of the zero tolerance. So, right, for dermo, right. So again, thank you, Ryan. Appreciate it.